Today I'm going to be presenting on my thesis work, data analysis and software design to assist researchers in choosing effective endogenous genes for CRISPR-A. Just in general, like stop me at any time if you have any questions about anything during the presentation. Just a little bit about me, because I think there are some people here who maybe I haven't met in person or wherever. My name is Julie. I've been with the lab for about 3 years. I joined for the last year of my undergrad, and I'm a master's computer science student doing the BSMS program, and this is kind of my final presentation for all of that. So the guiding goal throughout the course of my master's degree research was to work on projects to help gather and compile rules for endogenous CRISPR activation using data-driven techniques. To complete this aim, I worked on 2 main projects that I will talk about later in this presentation. But first, a little background. I'm sure a lot of people in this call know this, but CRISPR is this really cool tool available for synthetic biologists that provides a mechanism for targeting specific sequences of DNA in a cell. And so CRISPR activation, or CRISPR-A for short, is a specific application of CRISPR that allows us to activate genes. And so kind of how it works is we have a deactivated or dCas9 protein that's going to combine with a guide RNA or gRNA at an activator. And the guide RNA is designed by engineers, and it has a sequence complementary to a target sequence in the gene, and it's going to be basically where we want to send our complex. And so we usually like to send that somewhere in the upstream region, upstream of the transcription start site of, or TSS of the gene we want to activate. And so then that complex will bind, activate transcription, and increase the production of this given gene. Note that this here, the PAM site, or the protospace or adjacent motif, it's the sequence immediately next to the target sequence. It usually consists of 3 nucleotides, and it's required for the dCas9 recognition. So my particular work focuses on endogenous CRISPR-A, which is CRISPR-A applied to genes found natively in an organism's genome. So endogenous CRISPR-A has the potential to take advantage of and improve already existing metabolic pathways in the cell to possibly produce certain metabolites in response to stimuli. So that's kind of why we're looking at endogenous CRISPR-A specifically. CRISPR-A seems to be like more flexible than a lot of tools, because it can theoretically target anywhere in the genome, but there are a lot of strict rules which govern which genes are good candidates for CRISPR-A. So when we change the guide RNA to guide the protein complex to a different place, it can dramatically change how effective that CRISPR-A is going to be. So a big question is, how can we tell if a gene is a good candidate for CRISPR-A? So one thing we can do is we can leverage known rules. So prior work has revealed that things such as the gene's baseline expression, the distance from the TSS, where the complex will bind, and the PAM sequence can have an impact on how effective that CRISPR-A is going to be. So using these facts can help us choose candidate genes for CRISPR-A. But many of these rules have not been collected and combined into a single model that can be applied to multiple genomes. So that makes it a little bit difficult. Prior work that I've done has involved incorporating the distance from the TSS and PAM sequence into a scoring function, but it didn't incorporate things like the baseline expression. Another thing that we might want to do to find good candidates is to discover new rules. The rules that we have right now for what makes a gene a good candidate for CRISPR-A is not all of them. Otherwise, we would know like exactly how to use CRISPR-A and how well it would work with every gene, and we don't know that, so there's definitely more to learn. So we should be able to learn more through experimentation, data analysis, and probably machine learning. But a challenge with that is that we need lots of data, and it's also a good idea to have a really good understanding of that data and its format. So I'm a computer scientist. I love leveraging data to use to solve problems. And the data in biology is super rich, and analysis techniques can help us uncover information and reduce time spent just kind of doing trial and error to learn new things. And so the analysis part that I did in kind of my projects is part of the design, build, test, learn pipeline. Basically, it's a cycle where an experiment is designed, then like maybe it's built, like parts are built, then it's tested, and then we kind of learn from those tests. And so my parts are in the learn steps, where we're taking results of past experiments to learn new rules and then suggest future designs. So I worked on kind of 2 thesis projects, and these are kind of, that was kind of all the motivation for them. So uncovering new rules really motivates my first project, which I'm going to talk about shortly, which is mock data generation for FACS-seq CRISPRa. So for this project, I wrote a software pipeline that would generate synthetic or mock data that would be some similar format to an experiment that we were planning to perform. And leveraging and compiling existing rules motivates my second project, putida gene expression CRISPRa filtering. And for this project, I used existing putida gene expression data to filter a list of genes down to a smaller list of suitable candidates for CRISPRa. Okay, so I'll start talking about that first project, the mock data generation for FACS-seq CRISPRa. So I joined this project around the spring of 2020, where we aimed to perform an experiment to get basically different expression levels for different guides to kind of test their effectiveness. We plan to perform data analysis and machine learning techniques on this data to uncover more CRISPR rules. But due to COVID, the experiment was delayed. The lab that had all the devices that we were going to use kind of shut down. And so we weren't able to carry that out. So kind of to deal with that delay, the project that was proposed was to create synthetic data that mimicked the results we were expecting to get from the experiment in order to have a starting point for developing analysis techniques. So the experiment was going to consist of us testing 1,700 different guide variants with 15,000 replicates per guide. So many of those guide RNAs were going to be from the same library with just slightly different sequences. And we would measure the fluorescence, and that would be the protein or the product that was the gene that was being activated. And so that would be taken as a measure of the effectiveness. Then cells would be binned using this process called fluorescence activated cell sorting, or FACS for short. And so basically cells are binned on their fluorescence level. And then each cell in each of the bins would have a barcode added to it. And so that's just a short sequence that would be unique for each barcode. And looking at this barcode would determine if which bin that cell was sorted into. And so then kind of everything would get recombined, and then the populations would be sequenced to inform which guide was present in each bin. It was somewhat ambiguous what data was going to be produced because people on the project had not worked with the FACS process or the Illumina sequencer before. And so having synthetic data available could help with the development of analysis pipelines before the analysis was carried out. And so I designed and wrote software that would generate this synthetic data. So this is kind of an overview of the software pipeline. So here we have, these are the 2 kind of scripts that were written. And kind of an arrow indicates that this is an arrow to a script means that this is input data, and an arrow from a script indicates that this is output data. And I'll be talking about each of these scripts in more detail. So starting with the first script, which is the script called mock bins, which will generate a binned histogram of fluorescence expected to be output by the FACS process for various guides used in the experiment. So it takes in a list of all of the guides, and then it's going to output kind of a fluorescent histogram for each of the guides. And it will also output a file that has the kind of true parameters used to generate those histograms. The data created by this particular script is not representative of any real data that would be available for researchers. It kind of just is an intermediate step before the next program, which will, the next script will generate data that is more representative of real data that we would see. So the model that we used to kind of determine the fluorescent distributions was a Gaussian distribution. So for each of the 1700 guide variants, its fluorescence was modeled to come from a Gaussian distribution with a mean and a variance that were generated from a uniform distribution. So basically any values in between the minimum and maximum are equally likely for this particular variance, mean and variance. And so the uniform distributions can be specified by the user what they want the minimum and maximum to be, but they're defaulted to these values. They were kind of chosen through kind of just trial and error, looking at the distributions and kind of looking back at some real histogram data that we had and just making sure that it was kind of similar to that real data. So let's say for each cell, let's say that the guide variant in that cell is guide variant I, then its fluorescence will be a sample drawn from that normal distribution, that Gaussian distribution associated with that particular guide variant. So this is an example of just that first step. So this is an example for just a guide. So first we draw a random mean and variance from that distribution, again, because we don't actually know what the true mean and variance is going to be in the real experiment. So we're just coming up with random ones. And then we are going to draw 15,000 samples from that normal distribution. And we get a distribution like this. And so the one on the left and right are basically the same, except in this one, the x-axis is linear, and in this one, the x-axis is a log scale. And I believe that we did that because that's what the real data that we had looked like. So we were kind of just eyeballing it to compare. So the next thing to do would be bin kind of at a lower resolution. So the cell sorter is not able to record the exact fluorescence provided by a cell. It can only determine if that cell's fluorescence falls within a certain range. So the amount of resolution we expect the cell sorter to be able to distinguish can be determined by the user. I think because at the time I wasn't sure what it would be. I think it ends up being about having about like 96 separate bins. But in this example here, we just have 10 bins. And so the script will output two files. The first file contains the true mean and variance of each of those normal distributions for each guide. And the other file that's produced, mock bin counts, contains basically a data representation of those bin histograms that got generated for each of the guides. Okay, so the next script is called mock FACS-seq, and that's going to end up generating the FACS-seq files, which are representative of the real files that we expected to see from the sequencer. So it's going to take in the list of guides as well as the sequences for each of those guides. It's going to take in the file that has the histograms, the bin histograms for each of the guides. And it's also going to take in a file that contains short barcode sequences that will end up being associated with each bin. And we'll output these two FACS-seq files, one for read one and another for read two. Now, how we don't end up using the true parameters, that was kind of for any analysis that would be done in the future to kind of sanity check that analysis. Okay, so what is in a FACS-seq? So a FACS-seq is a type of file that contains information about sequencing, and basically every entry in that file contains 4 rows. The first is a sequence identifier, which has all this stuff in it, kind of all separated by colons. The second row is a sequence. The third row is just a plus, indicating that the next row is going to be an indication of the quality score. And so all items in these mock FACS-seqs are placeholders, except for the read number, which is going to be 1 or 2, depending on if this particular entry is read 1 or 2. The index is going to be the short barcode sequence associated with the bin. And the sequence is going to be a subset of the guide variant sequence, which is calculated differently depending on if it's read 1 or read 2. So the text on this slide is a bit specific and technical. So I'm going to give like kind of a really high overview of how we would go about generating this. And then I'll show how this works with an example. So for each of the guide variants from the mock bins counts file, so all of our 1700 guides, the first thing that we're going to do is generate read 1, then we generate read 2, and then kind of for each of the separate bins, we will kind of tag the barcode on to our entry, and then for however many entries are in that bin. So let's say that there's n entries in that bin, then we will output n entries with read 1 and n entries with read 2. So an example, let's say that we have this sequence. I'm not going to read the whole thing out, that would be silly, but it's this guy here. And so to calculate read 1, the first thing we need to do is find this 4-letter sequence TAGG. And then we're going to grab the 39 base pairs, including TAGG, and it's going to end up being that. And that's just what read 1 ends up being. For read 2, we're going to take the reverse complement, and we're going to find CCTA. And then we're going to take 36 sequences, including and ending with CCTA. So it ends up being a little bit shorter, but it ends up looking like that. So read 2 ends up being this guy. So let's say that we want to generate all the entries for the second bin. Let's say that there are 32 reads in it. And so we look at what barcode we have associated with the second bin, and that ends up being the sequence here. So then we're going to generate 32 read 1s, and they'll end up looking like this. So kind of everything here is a placeholder, except for this is going to be 1 because it's read 1. This is the barcode, and this is the read 1. And then the 32 read 2s are going to have 2, the barcode, and the read 2 sequence. And so kind of the results of the synthetic data, it ended up getting a total of 26 million records per file, which is huge. It took about 5 minutes to generate these FASTQ files on my laptop, and these end up being huge because they're super flat files. Ended up being 30 gigs each. Really hard to send over Slack. But it was reasonable to read through the files. So you could read through the files with BioPython in the order of minutes, and hopefully get that file structure down to something more compact, so you wouldn't have to always spend a few minutes looking through it, implying that this process could be done in a reasonable amount of time. So unfortunately, this data was never confirmed to be a good stand-in for real world data. We didn't end up generating this data with the FACS-seq process. It ended up being considered to be too time-consuming and tedious to set up, and so it was abandoned for amplicon sequencing. And so future work with this particular project would need to involve gathering real-world data and maybe even a small sample just to confirm the validity. And future work should also consider using the mock data to actually test those analysis pipelines and see if it was a good stand-in for real data. Okay, so I'm going to start talking about project 2, which is putida gene expression CRISPRa filtering. So putida is this cool little soil bacterium here, and it has a potential to be a great vehicle for creating various renewable materials. Putida CRISPRa has been demonstrated, but the rules to suggest effective CRISPRa are currently not available. And so kind of in this project, we aim to discover which endogenous genes were good candidates for CRISPRa. And so we wanted to use data sets and known rules to generate a list of suitable endogenous gene candidates for CRISPRa. So kind of one of the biggest focuses of this particular script that I ended up writing was to take into account the baseline expression of various genes. So in general, CRISPR-A is very sensitive to promoter strength, which is I'm going to use that kind of interchangeably with baseline expression. If a promoter is too weak, it will be difficult to activate and acquire fold change. But also if a promoter is too strong, then fold change due to activation will also be minimal. The range of kind of an acceptable baseline is not known, and so we need to infer that from existing data. And so for this project, we had various data sources that we were using. So we had this RNA Seq data, which is going to be data that we kind of used as a stand-in to measure the baseline gene expression of various genes in P. putida. We also had a list of activated genes. And I'll talk a bit more about these two data files in more detail in the following slides. But we also had a genome scale model outputs. And so this was a list of genes that were considered good predictions for effective CRISPR-A and CRISPR-I genes from a genome scale model. And we also had a set of data which indicated which genes were TSS primary promoters. And so I'll talk a bit more about those top two files. Excuse me. So the RNA Seq data is data that measures the baseline gene expression of various genes in P. putida, and it was provided by Joshua Elmore. It contains over 5,000 P. putida genes, and it has 4 columns. So it has a locus tag, which is an identifier that represents the gene that we're looking at. By convention, it begins with 2 capital P's followed by an underscore and then a 4-letter ID. It also has a name, which is a description of the gene's product, the raw read, and also FPKM, which is kind of going to be one of the important things. This is short for fragments per kilobase of transcript per million mapped reads, kind of a mouthful. But basically, this is the total reads for that sample divided by 1 million divided by the length of the gene. And this was used in the pipeline to represent the baseline gene expression. So it consisted of 14 CSVs that had 4 different experimental conditions. Kind of each of these boxes is a different experimental condition. So there were maybe not 2 different promoters, but kind of two different of these guys, and then 2 different nitrogen sources and 3 to 4 replicates per experiment. And a note on the nitrogen sources. So if it's an NH4, that means the nitrogen source was ammonium. And if it's NO3, then it's a nitrate. And the experiment with the ammonium as the nitrogen source was going to be closer to kind of what we wanted to do. And so in the script, the user can specify to only use these data sets in the filtering analysis. Kind of the other super important data set was the list of activated genes. And so these are 11 genes tested to see if they were suitable candidates for activation based on their baseline expression. And 4 of those, the ones highlighted, were found to have suitable activation. And this experiment was performed by ICE. So I'll kind of detail the steps in this pipeline. So number 1 is normalization. So kind of for each of the data sets, the FPKM was normalized by the FPKM of a reference gene. So we would kind of grab the FPKM for this gene in PII and then divide every other gene, every other gene's FPKM by that particular FPKM. The next thing we would do is average across the results. So basically, there would be basically 1 data set per experimental condition. So if we were looking at all of the data, we have those 4 different experimental conditions. So we should end up having 4 data sets. The next thing that we might want to do is filter by the primary TSS, just removing any genes that are not in the primary TSS data set, because we know that those primary TSS genes are better candidates for CRISPR-A. The next thing is to filter by activation. And so we want to filter genes that have activation within the desirable range. And so we're going to look at those genes that we found to be suitable and kind of use them as a reference. But a big question emerged at this point: how do we do this when we have multiple data sets? Do we kind of filter separately for each data set? Do we figure out what a global maximum minimum are that we want to have the baseline ranges be within for every data set? If a gene gets filtered out of one data set but not another, is it still considered a suitable candidate? Or does it need to survive the filtering process in all of the data sets? So to answer this question, we performed a data analysis experiment. So basically, we would try each filtering method and then count how many of the 4 genes considered suitable candidates were kept in the data set and how many of the genes that were considered not suitable were thrown out. And the assumption here is that the method that keeps the most suitable genes and throws out the most unsuitable genes is going to be our winner. And so here are the experiment results. There were, I guess this is several different experimental conditions. I think this ends up being 9 different conditions. I'm bad at counting. I'm happy to talk more about this, but kind of the big takeaway is that the second one here is the winner. So this would be using a maximum and minimum range. So grabbing the range for each data set. So kind of looking at what the RNA Seq FPKM was for these 4 genes, and then using the max scene and the minimum scene as the max and the min of the range we would consider suitable. And doing that for each data set. And then we would require the gene to basically survive the filtering process for all of the data sets to be considered suitable. And so this kept all the 4 genes that were suitable candidates, and it threw out 4 of the 7 genes considered to be not suitable, which was kind of the highest thing there. So the results of this whole process, at this point, the software reduced the over 5,000 genes down to 503 genes, which, cut out 91% of all of those genes. For CRISPR-A, 15 genes were also provided by the genome scale model. And when we kind of took the intersection of the genes produced by this script and our script, we got 5. So a much smaller list that we could look at. And the pipeline finishes running in under 1 minute, which is pretty reasonable for this type of analysis. So some final thoughts on this project. This work, kind of going back to the beginning, is meant to be part of that learn and design part of the design, build, test, learn cycle. But these, as far as I know, these chosen genes have not been validated yet. And so future work should continue that cycle and validate those and reassess and design the software to incorporate any new insights that we gain in the next iteration of the cycle. Another kind of big limitation of this project is the validity of the filtering analysis method, not necessarily founded in statistics. It was based off of kind of a model where we're making these assumptions that we wanted to keep genes considered suitable and throw out ones that were unsuitable, but not super statistical there. And again, very hard to verify without any validation data. Okay, so kind of repeating that, biggest limitation, you don't have a design, build, test, learn cycle if it just kind of stops. Future work needs to validate these approaches so the cycle can continue.